The overall goal of this experimental method is to enable real-time study of intracellular signaling using FRET imaging of cells embedded within three-dimensional hydrogels that mimic natural tissues.
This method can help answer key questions in the field of developmental biology, pharmacology, and regenerative medicine, such as development of optimum biomaterials and high-throughput drug screening in engineered 3D microtissues.
The main advantage of this technique is that FRET can be used on a large number of cells in numerous hydrogel types using conventional wide-field microscope systems.
After fabricating molds for hydrogel casting and suspending cultured cells in hydrogel solution according to the text protocol in a tissue culture hood, add the cell-containing precursor to the casting molds using sterile technique. Prior to cross-linking, centrifuge the cells suspended in hydrogel precursor to optimize imaging by confining cells to a single focal plane and minimizing the out-of-plane signal. Next, to cross-link PC hydrogels, use a UVA light source for an exposure time equal to 3.2 joules per centimeter squared per millimeter of thickness to irradiate the cell-containing precursor.
Alternately, use a less expensive handheld dental lamp. Avoid overexposing the hydrogel because this will decrease cell viability. Physically cross-linking hydrogels are instead placed at 37 degrees Celsius.
After cross-linking, remove the PDMS hydrogel casting mold and replace it with the previously prepared